Today we're going to start talking about proteins. Proteins are one of our big class of macromolecules, and they're extremely important in biology. You're going to be hearing about proteins in many forms, but especially in terms of what we call enzymes, which we'll talk about in a later presentation. Today I want to make sure you understand the foundations of proteins, kind of the pieces that help fit them together, the different monomers, and what proteins do and how they're structured. Before we do that, however, let's review yesterday's presentation. Yesterday we talked about DNA and RNA molecules, the nucleotides, nucleic acids. And so here's a couple questions that I would like you to try answering. What I want to do, I'm going to propose the question to you. I want you to stop the video or pause it, then try to answer the questions on your own, and then come back and I'll walk through the answers. So go ahead and pause as you need and see what you can do based on our conversations yesterday. So our first question here, compare RNA and DNA with respect to the following aspects of their structure: sugar, bases, number of separate strands, and three-dimensional structure. And then finally, draw the complementary base pairing that occurs in DNA. What nucleotides pair up with what nucleotides? And be sure to indicate the hydrogen bonds that are appropriate. Okay, go ahead and pause the video now, and as soon as you've answered these to the best of your ability, come on back and we'll discuss the answers. Okay, let's go ahead and start breaking this down into DNA and RNA. So DNA versus RNA. In terms of sugars, our first category, sugar, DNA has deoxyribose, while RNA has ribose. If you were to get into the chemical structure of these two different sugars, deoxyribose has one less oxygen than ribose has. Otherwise, they're structurally very similar. The bases. DNA has adenine, guanine, cytosine, and thymine, while RNA has adenine, guanine, cytosine, and uracil. So they contain the same bases except for thymine in DNA and uracil in RNA. The number of strands. DNA is a double-stranded molecule, while RNA is a single-stranded molecule. And the fourth one, three-dimensional structure. DNA is what we call a double helix. It has the two strands, and those two strands twist around each other, forming almost like a spiral staircase. RNA, on the other hand, is single-stranded, but because it's one-stranded, RNA is actually able to kind of bend back and and base pair with itself in certain areas, and forms all kinds of unique shapes. And finally, draw a complementary base pairing that occurs in DNA. Well, if you look at the base pairs that occur, we have adenine, guanine, cytosine, and thymine. What pairs with adenine? Thymine does. What pairs with guanine? Cytosine. Cytosine, guanine, and thymine, adenine. The bonds that form between the bases are hydrogen bonds, and adenine and thymine get 2 hydrogen bonds between them, while guanine and cytosine get 3. So that's our basic bonding pattern. Okay, let's go on to the next review slide. A double-stranded DNA molecule. The parts of a DNA molecule are important. So let's review some of the key characteristics here. First off, the monomer, the basic building block of a DNA molecule, is the nucleotide. This is a nucleotide. A nucleotide always includes a nitrogen base, a deoxyribose sugar in DNA, or a ribose sugar in RNA, and a phosphate group. These are the 3 pieces that are in every nucleotide. Nucleotides are joined together to form a chain. So this is a chain here of nucleotides. You can see nucleotide 1, 2, 3, and 4. And these nucleotides are bonded together with covalent bonds using condensation reactions to bond them together. The next part we want to look at is the bases. So this is a nitrogen base, this is another. Well, the bases are bonded together with the other strand. And they are what are called complementary bases because A's and T's join and C's and G's join. So if you know one side, you automatically know the other side. And the nitrogen bases are hydrogen bonds, which are much weaker than covalent bonds, much more easily disrupted and broken apart, which is important because in the DNA molecule, we will routinely take our two strands and rip them apart and bring them back together. We don't want to spend huge amounts of energy. However, even though a hydrogen bond is weak, you can see right here that one DNA molecule, double-stranded, will have many hydrogen bonds in the order of millions, okay, or hundreds of millions possibly. So many hydrogen bonds, each are weak individually, but together they form a fairly strong connection. And the final thing I want to really point out here is that our DNA strand is what we call anti-parallel. If you look at our DNA strand on one side, so let me erase these, our DNA strand here on the right-hand side has 5' up at the top and 3' in the bottom. Another way of looking at that is the nucleotide here has the phosphate up here at the top. The nucleotides all have the phosphate at the top. If we go to the other strand, it is reversed. 5' end is down here at the bottom, 3' is at the top, and if we look at each nucleotide, the phosphate is headed downwards, not upwards. This is called anti-parallel. Okay, if I'm using my fingers to show this, this is a nucleotide, here's my phosphate, here is my sugar, here's my nitrogen base, and here's my other nucleotide. The nitrogen bases join, but this would be incorrect. To make it correct, I would rotate one of them so it points down. This is how the nitrogen bases bond together in our two strands. Anti-parallel. They are parallel, they run together, but they have opposite sides. The 5' is on the opposite direction on each of them. This will become very important when we start talking about copying DNA molecules or reading them. Transcription, translation, DNA replication. So we're not applying that fact that they are anti-parallel right now, but it's important that you know that. I also want to remind you that on the first test we have over chapter 3 dealing with DNA molecules coming up, I believe, Thursday of next week, you will be asked to draw a DNA molecule. You will need to draw it with this level of detail, and you will need to be able to label all the different parts of the DNA molecule. Covalent bonds, covalent bonds, hydrogen bonds. You need to know what makes up a nucleotide, phosphate groups, sugars, the bases, the structure of different bases. The only thing on here that you would not have to include is when you look at the hydrogen bonds here. A and T have 2 hydrogen bonds, C and G have 3. That's important. But you would not need to include the oxygen, the NH and the O. So you could cross that part out on each side of the hydrogen bonds. You still need to know 2 bonds or 3 bonds, but what's on either side of the bond, you would not need to know. My best recommendation for learning how to draw these things efficiently is every day between now and the test, draw a DNA molecule. Start off looking at the DNA molecule in your book and then drawing it. And over time, start trying to draw more and more without looking in your book. If you do this every day, by the time the test comes, you should be very efficient at drawing this molecule, and you'll do really well on that part of the test. It'll be an easy set of points for you to get. Okay, so make sure to do that. Okay, another review question. DNA replication and transcription. DNA is constantly being copied either into another DNA molecule or being copied into an RNA molecule. So here, the DNA sequence below is one of 2 strands in a double helix. Write the nucleotide sequence for each of the following, making sure to label the 5' and 3' ends of each molecule. So it wants you to write the complementary DNA strand, the strand that matches and connects to this strand. It wants you to write the RNA strand that's transcribed from this DNA strand. And then finally, the RNA strand that is transcribed from the complementary DNA strand. Okay, so With that in mind, pause the video, try to come up with each of those three, then come back and we'll revisit and see if we all got the same answers. Go ahead and pause. Okay, so let's start off. Let's go in here and do these in order. So the first one, complementary DNA strands. So this is my DNA strand that I'm getting. Now remember, they are opposite, anti-parallel. So that means this is going to be 3' and this one over here is going to be 5'. So those are my two ends. And now the base pairing is very simple. What matches with this original strand? Well, A bonds with T, G, C, C, G. So I'm just going to keep going. T, A, C, G, A, T, C, G. Oops, I'm running out of space. I need to erase my ends here. Okay, let's see, where was I? C, A, T. That's my 5' end. That would be our complementary DNA strand, so number 1. Next up, the RNA strand transcribed from the original strand of DNA, right here. I'm going to do that here. I'm going to do that with our RNA strand. I'm going to do it here. So that would also be 3'. Okay, well, 5' on the other end, but I'll draw that in at the end when I have space. This is going to be done almost the exact same way we did this first DNA strand. We're going to do complementary base pairs with one difference. RNA does not use thymine, no T's. Instead, it will put down a uracil where a thymine should have gone. So as I go through here, I'm going to end up with U, C, G, U, A, C, G, A, T, oh, no, T, U, C, G, C, A, U. And this is 5'. Hopefully you got that. Next, the RNA strand transcribed from the complementary DNA strand. So that's saying, what will we get RNA if we went from this strand to RNA? So here, I'm going to use this one to copy, and the 3' end, so I'm going to match, this is my next RNA strand, 5', and I'm just running through, so I'm going to end up with A, G, C, A, U, U, G, C, U, A, G, C, G, U, A, 3'. Okay, and that's how you do it. So you will be doing lots of these complementary DNA strands or an RNA strand and stuff, so make sure you understand how to do it. Hopefully you got those correct. If not, make sure to ask the questions during class. One more review for us. RNA. So far we focused mainly on DNA. What about RNA? So copy the strand of RNA below, then find 2 complementary sequences that are 4 base pairs in length and underline them. Then draw the results of the molecule with those 2 sequence base pairs to each other. Include the individual bases in your drawing. So go ahead, pause the video, see what you can do, and then come back when you're ready to look at the answer. Okay, how'd you do? If you look really carefully here, what you're going to see is UUCG and CGAA. Those are our 2 complementary sequences. Not whole strands, but sequences. Now, RNA molecules, because they are single-sided, they are a one-sided molecule, are capable of bending back and bonding to themselves, like you see in this little drawing. Now, I'm not going to draw out every nucleotide here, but what we would have in this particular case with this molecule, because of this matching base pair, complementary base pair, my molecule here would go 5', it would start coming here, would loop around, and then it would bond to itself. So we'd actually get 4 complementary base pairs matching, and then it would go over here, and this would be our 3' end. You, of course, could draw that in more detail. You could add the actual nucleotides. But because of this, the fact that it can bend around and match to itself, the RNA molecules can make many unique shapes, and sometimes those shapes are very important to what the RNA molecule will do, as you'll see in future lectures. Okay, that's enough of a review. Hopefully DNA and RNA are making sense to you. Now I want to start jumping into our main topic for today, proteins. Let's take a look at proteins. Proteins are polymers. They are complex molecules, one of our macromolecules, and they serve a bunch of really important roles in your body. A small sampling are proteins can be enzymes, talk about those coming up, defensive proteins, part of your immune system. They can be hormones or regulatory proteins. They can be receptor proteins, receiving signals. They can help store things. They can be part of what your cells are made out of, structural proteins. Transport proteins that help move things around the cell and between cells. Or they could be genetic regulatory proteins. So you can see, proteins have a lot of jobs in your body. In fact, in each of your cells, you have more than 100,000 different types of proteins. So this is a very diverse group of macromolecules. And like other macromolecules, proteins are made of monomers. So remember, a polymer is a large molecule made of small monomer building blocks. In the case of proteins, the monomer is an amino acid. So amino acids are the building blocks of proteins. And if you think back to that activity we did with matching biochemical molecules, this is the defining characteristic of an amino acid. On one end, on the left-hand side, we have an amino group. That's this N plus with H3 connected. That's the amino group. We then have a carbon in the middle, and then we have the carboxyl group. Now this could be COO minus or COOH. So sometimes we lose the H and we end up with a negative charge there. Sometimes we keep the H. So you can see it either way. And then finally up here we have a hydrogen attached. All amino acids have this set. This is the same on every single amino acid. It's the R group, the side chain. The functional group is another term you'll hear. This is what makes amino acids different from each other. There are 20 amino acids that occur extensively in the proteins of all living organisms. These are called the essential amino acids, and right here you can see a chart showing them. This chart is taken from your textbook, page 43. So if you want to ever go back and look at it, you can. Now, got some good news and bad news. The good news is you do not need to memorize all these functional groups. That used to be required in AP biology, no more. So thank goodness we don't have to memorize all these. But you do need to understand how the chemical properties of the different amino acids affect the final protein. So what I want you to look at carefully here is see how these amino acids are categorized. The ones up here in blue are all electrically charged. Some of them are positively charged, and some of them are negatively charged. That is important because the R groups, if you are negative or positively charged, you interact with each other and your local environment in different ways. For amino acids, most of the time the environment we're talking about is water. Water is, the inside of the cell is about 70–75% water, and so proteins are constantly coming into contact with water. And the charge on an R group can influence how it reacts with water. Molecules that are positively or negatively charged are what we call hydrophilic. Hydrophilic means they love water. A hydrophilic molecule likes water. It is attracted to water because water is a polar molecule that has an electrical charge. Okay? Actually, it doesn't have a cumulative electrical charge, but it has one side of its molecule is slightly negatively charged, another part is slightly positive. So positive and negatively charged amino acids like to interact with water. Okay, also we have some amino acids, so our next group, B here, that are uncharged, but they're polar. Okay, the group they have here is a polar molecule, their R group, and therefore these are also hydrophilic. They like to interact with water. Of course, we also have amino acids that have non-polar R groups. These are hydrophobic. They repel water. They are not attracted to water. They, in fact, fear water, hydrophobic. So again, depending what chemical properties different amino acids have, that influences how they react with water, which in turn can influence the shape of the protein. And the shape of the protein is of the absolute most importance when we're talking about protein function. So in this case, what you have to be aware of is that there are times mutations and various changes happen where one amino acid will be substituted for another. If it is substituted for an amino acid that has a very similar chemical property, for example, a positively charged amino acid like arginine, right here, is replaced with histidine, both of which have a positive charge, that will have a minimum impact on our finished protein. Because it still, even though it has a different amino acid, it still shares the same chemical properties. In contrast, if we have serine, a polar hydrophilic amino acid, is replaced with methionine, which is a non-polar hydrophobic amino acid, that is going to have a more pronounced impact on my protein because these have very different chemical reactions to water, and therefore it will affect the shape of the protein. It could affect how my protein functions. Okay, so you don't have to memorize the amino acids or the amino acid functional groups. Don't worry about that. But understand, if you see a question telling you about an amino acid and it tells you its chemical properties, hydrophilic, hydrophobic, charged, polar, nonpolar, you need to understand that if you change that for something with a different chemical property, that will affect the function of the protein. Okay, so we have all these functional groups. Let's start talking about how amino acids become proteins. Amino acids can be linked together with covalent bonds that we call peptide bonds. Proteins are often called peptides, polypeptides and such. So anytime you see the word peptide, think proteins. So here I have two amino acids, and I want to join these together. What I'm going to use what is called a condensation reaction, just like we saw with DNA and nucleotides. So condensation reaction, RXN, short for reaction. And I'm going to join the carboxyl group on one amino acid with the amino group on the other. From the carboxyl group, I'm going to remove the oxygen. From the amino group, I'm going to remove two hydrogens. These are going to join together and form water. That's why it's a condensation reaction. Water is being formed. And after the water breaks off, my carbon from the carboxyl group and my nitrogen from the amino group will form the peptide bond. And now these two amino acids, number 1 and number 2, are joined together in a chain. And I can continue doing this repeatedly, creating a long chain of amino acids. How long? Well, some are what we call oligopeptides. These are relatively short. They contain 20 or fewer amino acids. And you'll rarely hear us talk about those because polypeptides, poly means many, polypeptides are much more common, and these are longer, from 51 up to 30, almost 35,000 amino acids, huge numbers of amino acids. Proteins are huge molecules, and they can be constructed of tens of thousands of amino acids to form the final protein. So a protein is formed by sequential addition of amino acids. So it's many amino acids linked together will ultimately form a protein. And each amino group of the amino acid reacts with the carboxyl group of the previous amino acid in a condensation reaction, ultimately forming long chains. And we end up with a polypeptide chain. So here you can see we have a chain of amino acids bonded together from condensation reactions, held together by peptide bonds, and each circle here of a different color represents a different amino acid. The sequence of amino acids can be random or appear random, but it's actually the sequence is very, very important for the development of a particular protein. Different proteins will have different sequences, but any given protein will always have the same sequence. So we get a polypeptide chain like this. It's not actually a protein yet, it's still a polypeptide. The next step would be to modify this polypeptide chain and form it into a very unique particular shape. For example, this one. This is catalase. Catalase is an enzyme protein that is commonly used to break down hydrogen peroxide. And if you look at this molecule, you see this is an incredibly complicated molecule. There are many, many unique twists and turns in the chain of amino acids here that result in a complexly shaped molecule. And that shape is vitally important to the function of a protein. If it does not have the right shape, proteins will not work. They do not function. So this shape is required before we officially call it a protein. So polypeptide hasn't been formed into shape yet. Protein has formed into shape. So how do we get the right shape? Well, protein shape, or protein structure as we commonly call it, is composed of 4 different levels: primary, secondary, tertiary, and quaternary structure. And they occur in order, and very particular things happen in each. So let's take a look at these one at a time. Primary structure is the simplest and first layer of structure in a protein. Primary structure is simply the precise sequence of amino acids in a polypeptide chain. So here in the picture, and this picture is out of your textbook, you can see this is an amino acid. Here's the next amino acid, and the next one, and the next one, and so on. It's a chain of amino acids. And it might be arginine, then serine, alanine, cytosine, and so on. We commonly abbreviate amino acid names with just a single letter, like in this case, RSAC. Sometimes it corresponds with the first name of the amino acid, and other times it does not. You do not need to memorize the abbreviations, just understand they commonly use them. And they can't always correspond with the name because there are 20 amino acids and sometimes multiple ones. Arginine and alanine here start with the same letter, so they still have to distinguish. So primary structure is simply which amino acids, in what order, and how many. That's primary structure. Secondary structure is a little more complicated. After you get primary structure, secondary structure forms in 2 very unique shapes that commonly occur in proteins. So these are regular repeating spatial patterns in different regions of the polypeptide chain. The first one, right here, is what we call an alpha helix. An alpha helix is when you have your DNA sample, your protein sample, polypeptide, and it begins to wrap in kind of a corkscrew. An easiest way to visualize this is if you have your finger out and you have a string that you start wrapping around your finger. That would look like an alpha helix. And here's a drawing of it on the left-hand side. This is an alpha helix. Alpha helixes are not the entire length of the polypeptide. They are a short section of it that forms this helix shape. And the helix shape is held together generally by hydrogen bonds between different portions of the R groups. The second form that forms is what's called a beta-pleated sheet. A beta-pleated sheet occurs when your polypeptide chain goes and it doubles back, and there is an interaction between the 2 sides, kind of like what we saw in RNA molecules. So when you get this doubling back, here we have the 2 strands, and you get some hydrogen bonds forming between the 2 groups, the 2 different parts of the strand, and that is called a beta-pleated sheet. Again, it does not occur over the entire section of the polypeptide, just short segments of it. So the formation of alpha helixes and beta-pleated sheets is what we call secondary structure. Now, any given protein might have no alpha helix and beta-pleated sheets, might have one, or might have multiple of them. Each protein is different. Once we have secondary structure handled, we move on to tertiary. Okay, look at our diagram here, and what you can see I've got a polypeptide chain stretched out here. Portions of it have alpha helixes. Looks like we've got 2 alpha helixes on this particular one, and then 1 beta-pleated sheet that formed right here. So this one has already gone through primary and secondary structure, but now it's time for tertiary structure. Tertiary structure is determined by the amino acids in the polypeptide, how they interact with each other and with their surrounding environment. This results in a distinct 3-dimensional shape for our protein. So what I mean by that is the R groups, remember we talked before, R groups have positive and negative or polar and nonpolar sections in them. Well, positive and negative R groups are attracted to each other. Positive and negatives attract. And so R groups that are opposite, positive and negative, will be attracted to each other and will actually pull closer together, forming a kind of ionic relationship. On the other hand, two positively charged molecule R groups will repel each other. They'll push away. Polar and nonpolar molecules don't get along, so they will move away from each other, while polar and charged molecules will move closer together. So as they interact with each other, they push each other away or move closer to each other, and you start getting a distinct shape appearing. Now add the fact that this protein is floating around in your cell where there is water, and these groups interact with water, so that will continue to modify the shape. Positive, negative, and polar R groups like interacting with water, so they will be on the outside of your protein near where the water is. Nonpolar R groups, on the other hand, are repelled by water. They are hydrophobic. They do not like water, and so they will seek shelter in the middle of the protein, surrounded by the other groups so that they can avoid the water. So as you can see, this very quickly starts giving us a very unique and pretty complicated 3D structure for our protein. This is shaped by the different functional groups that are present and how they interact with each other and interact with the surrounding environment, particularly water. So that's primary, secondary, and tertiary structure. We have one more, quaternary structure. Many proteins are made of multiple polypeptide chains. So after primary, secondary, and tertiary structures are finished, multiple polypeptide chains will come and join together to form our quaternary structure, the final protein. Here you can see 4 different subunits or polypeptides, 1, 2, 3, and 4. They did them in different colors that have come together, and each with their own unique shape, and they have bonded together using hydrogen bonds and other types to form our final finished protein. Okay, that now, after going through all those 4 different sections, primary, secondary, tertiary, and quaternary structure, we have ended up with a complete protein that is now functional and has a unique, important shape that allows it to carry out its function. Be aware that protein structure can change. It is not permanently set. Increases in temperature can change a protein structure. As temperatures increase, we start to get a rapid movement of molecules that can break hydrogen bonds. Okay? And these cause hydrophobic interactions to occur. The magic number for most proteins is around 55 degrees centigrade, also 55 degrees Celsius, depending on what word you're used to. So if we increase the temperature too much, we start damaging proteins. So we have to be careful about increasing temperature. Some proteins can resist high temperature, but most of them cannot. Also, as we change the pH, we can affect a protein structure. pH, as acid and base, it actually has to do with the concentration of hydrogen ions. Acids are any molecule that add hydrogen ions. A base is anything that removes hydrogen ions. That's the chemical definition of an acid or a base. Well, proteins are generally designed to work really well in certain pHs. For example, the enzymes that are in your stomach right now work really well in a very acidic environment. But if you take them out of that acidic environment and put them in a different environment, they might not work very well. Similarly, other enzymes might work really well in a basic environment. And if you put them into an acidic environment, they won't work very well. So each protein is optimized to work under certain pH conditions. And the more you deviate from those conditions, the less well it works. Okay, because the protein begins to change shape. It cannot maintain its shape. And when it can't maintain its shape, it ceases to function as well, or not at all. Other things that can affect protein structure. If you put it in the presence of a lot of polar substances act like charged molecules, and that can disrupt hydrogen bonds, can interfere, which can affect the protein structure. And similarly, if we dump a whole bunch of non-polar substances in there, that can also affect how the different parts of the molecule are interacting with each other and can change the shape of the protein. The reason I'm bringing up all these changes in shape in protein is because there's a really important concept that shows up on the AP test multiple times every year: denaturation. Proteins are very important molecules, and as you can see here on the left-hand side, we have a protein that has a very unique shape. You can see some alpha helixes, beta-pleated sheets in there. You can see twists and curves and everything that are tertiary structure. There might be quaternary structure if this was actually two different molecules. Can't really tell right now, but we have a protein that functions. And remember, its function depends on that shape. If you change conditions for a protein enough, something called denaturation can occur. Denaturation is when the shape of a protein is destroyed or ruined. So denaturation means we have lost the shape. Something has come in. Could be high temperature, change of pH, introduction of polar or nonpolar molecules. There's lots of possibilities. But anytime that happens and the protein loses its shape, you can see over here on the right-hand side, we have denatured this protein. We no longer have our alpha helixes. They have disappeared. We've lost our beta-pleated sheet. We've lost our unique 3D structure from that tertiary and quaternary structure. We just have a chain of amino acids here. When that happens, this protein is no longer able to function. It no longer holds the right shape, and it can't do its job. So denaturation can have catastrophic effects in a biological system in your body. If proteins that are doing important functions denature, they no longer function, and you are moving forward without those proteins. And that can be a really big problem. You will hear about denaturation multiple times, particularly in the contents of enzymes, which we'll start talking about later this week. But denaturation frequently shows up on the AP test. And they give you a hint, like an extreme temperature or something, and you need to piece together, denaturation occurs there. Protein stops working. What impact will that have? So be aware of denaturation, things that can cause it, and ultimately what the ramifications of denaturation are. Okay, I'd like to end by talking about an experiment that was in your book, just to make sure you understand how to read an experiment, what's going on, and what they learned from it. So this particular experiment kind of looked at the idea of denaturation, what levels of denaturation get messed up, what levels of structure get messed up by denaturation, and then finally, is denaturation permanent? So what they did, they took a protein, and protein working fine, and then they added some chemicals that denatured the proteins. Okay, ribonuclease is the name of the protein. Here, you can see it. Okay, and they denatured it chemically. And when that happened, we went back to primary structure, just the sequence of amino acids in a row that gave us our original protein. So basically unfolded the polypeptide chain. Then they removed the chemical that denatured it and waited to see what would happen. Would the enzyme start working again, or would it not work again? That was the question they were asking. So here's the results. What I'd like you to do, go ahead, pause for a moment, and try to take a look at this graph as well as analyze the data. See if you can figure it out, and then come back and we're going to talk about it. So go ahead and pause now. Okay, let's go ahead and talk about what happened here. So we have a graph, and they talk about disulfide bridges. Disulfide bridges are chemical bonds, kind of like hydrogen bonds and others, that are really important in the shaping of our protein. Okay, the ribonuclease in this case. Initially, the disulfide bridges were eliminated. That caused our denaturation. Okay, so at time 0 is right when we took away our denaturation agent. And we ask what happened when we did that. Okay, we're looking at two things. When did disulfide bridges start to form again, or did they? And second, when did the enzyme start working again? So let's look at A. At what time did disulfide bridges begin to form? Well, if we look at this, our first one forms again right here. We're talking there, this is in minutes. We're talking there probably in the first 5, maybe 10 minutes, we start getting disulfide bridges. And as you can see, as we go up the curve, more and more of them form. At what time did enzyme activity begin to reappear? Enzyme activity didn't really start appearing until right here. About 100 minutes in before we really started getting any enzyme activity at all. So why? C asks, why is there a difference? I mean, disulfide bridges are forming, why don't we have the enzymes working again? Well, remember, when those bridges form, disulfide bridges, that's not at the very beginning or at the very end of our process. Our protein still needs to go through secondary, tertiary, and quaternary structure. Remember, primary structure does not get damaged by denaturation. Primary structure is held together by covalent bonds, which are really strong. Increasing the temperature, changing the pH does not break our covalent bonds. But it does damage our hydrogen bonds, disulfide bridges here, and any other small, weaker bonds that affect secondary, tertiary, and quaternary structure. So in this case, once we remove the chemical agent causing denaturation, the disulfide bridges start to form again, but we have to finish secondary, tertiary, and quaternary structure before the protein, the enzyme, can work again. And that takes time, as we see here. The other takeaway from this is to understand that denaturation is not permanent. If you remove the conditions and give the enzyme enough time, it generally will start working again. However, it is non-functional until it's able to repeat primary, secondary, tertiary, and quaternary structure and reform the shape it had originally. And that does take time, sometimes extended periods of time. So denaturation is damaging to proteins. You will see that again, so please remember it. And understand that the reason it damages proteins is it changes their shape. So the protein no longer holds its shape, which is critical to its function. That wraps it up for proteins. Hope you learned a lot there. If you have any questions, make sure to bring them with you to class. Take care, and I'll see you next time.